Hello everyone, my name is Shahed Alsawaf and I'm currently doing my master's in molecular biology at Uskudar University, and today I will be presenting our study which is about exploring the potential of drug repurposing against abnormally regulated cell matrix interactions in the treatment of pancreatic cancer, which I am working on under the supervision of my dear professors, Professor Tuba Bal and Professor Zihni Onur Çalışkaner. Whenever we say pancreatic cancer, what comes to mind is its aggressiveness and the poor prognosis associated with it. Being one of the most hostile cancer types, it shows no or little response to the shown applicable therapies, such as chemotherapy, radiotherapy, and surgery. Its great survival rate is actually associated with the quality of tumor microenvironment, suggesting that it's best to focus on the cellular interactions underlying cancer to tackle it properly. That being said, the extracellular matrix and its involvement in cancer or the cellular survival pathways of both cancer tissues and normal tissues. So the importance of extracellular matrix is great that we can consider it as an option for treatment. So the main two interactions that are involved in pancreatic cancer are basically collagen interactions and hyaluronic acid interactions. But before we proceed with our presentation, I would like to highlight the importance of this study. Although being computational, yet it will be beneficial in setting the basis for new novel treatments, and therefore giving hope to patients and their families, of course. In this presentation, light will be shed on mainly discoidine protein receptors 1 and 2, which are involved in collagen interactions, to be able to investigate the new promising inhibitory agents to their functions. So we have two types of DDR proteins, and we can see the figures of their 3D structures as we see here in the slide. And we obtained, of course, these figures from a PDB database. Their function can be named as, in short, their involvement in cellular proliferation, differentiation, and matrix remodeling, as they bind to collagen and get activated by it. So the collagen presence and actually the collagen binding site in these proteins is very important for the activation of this protein and the function of it, of course. The steps I followed were, in short, data collection about most important promising drugs used to inhibit target receptors. Then, based on the data collected, ligand libraries were downloaded from Zinc-15 database and processed in order to prepare them for screening with the protein of interest. After studying the results of screening, molecular docking was performed to obtain the final results and to be able to visualize the ligands of best inhibitory ability. In this presentation, I will be focusing on DDR2 as an example for the detailed explanation of steps. So although I performed the same process for DDR1 and DDR2, I will be showing the details of this in details, of course, the steps. I will be showing them for DDR2 only. So, we collected information about promising drugs and their use. We focused on some parameters such as their molecular weight, their log p values, and their charge, since these parameters will be helping us in specifying the scope and range selected for ligand library download. In the bottom table, we can see the values which we chose the range to be between 450 and 500 daltons regarding molecular weight, and log p value as between 2 and 3. The molecular weight is relatively high. However, we do not care about that because our proteins have their own extracellular regions for them to bind, so the protein doesn't have to enter the cell actually. So therefore, if we use large ligands, it's okay to be considered in this study. As explained previously, the two ligand libraries were downloaded from Zinc 15 database. Then, the ligand libraries were processed and executed by using Windows PowerShell program to have them prepared to be screened in further steps. So we see here the first ligand library having log p value of 2 and the second ligand library having log p value of 3. So we have two ligand libraries, and we will also have a ligand library including only the cancer drugs such as dasatinib, imatinib, nilotinib, and pinotinib. And we will be showing also the results of them and compare all the ligand libraries together. Here we can see the steps of protein download from PDB database, then the processing step using Discovery Studio, where water, heteroatoms, and ligands were removed, along with, as we see here in the figure on the right side, we see the tripeptide of collagen. So we also remove this. We only keep the protein itself with no water molecules, no heteroatoms, nothing at all. Then we start the steps of virtual screening. As we see, we import the protein of interest first, and then we set it as an autodoc macromolecule. Then we import the ligand library we want to work with in the second step, as you see. Then we choose the grid box. Here we did a blind screening, since we covered all the protein, not only a section of it. Blind screening was performed, and then the results were collected. As we see here, binding affinity is the part we focus on. So binding affinity was important for us in this study. So we collect the results, and then we obtain the tables, and we sort them based on the binding affinity. Then we start with the steps of molecular docking, which is shown here. So first of all, macromolecule is imported and then processed by what? By removal of water molecules and also the addition of polar hydrogens. And the other processing step for protein molecule, which is the addition of Coleman charges and the spreading or the even spreading of it along the protein molecule. So first of all, we add the Coleman charge, then we try to spread it. So we have this, the one with the arrow, so spread charge in a deficient way. So we spread the charge, and then we make sure that it's completely or evenly spread on the protein molecule. The other steps of ligand addition, now we add the ligand, and the grid box will be selected. After we add the ligand, of course, the protein or the processing step of protein is also directly or automatically done on the ligand itself. So then we start with grid box selection. Here we don't perform blind docking. We only perform docking on the that is covering the section of collagen binding site on the protein. And then after we select the grid box and we run the autogrid steps, we perform autodoc steps, which one of the most important sections of it is selection of the genetic algorithm. And here we set the number of GA runs as 150 and the population size as 300 to obtain the best results possible. After docking is done, now we're done with the docking, the file is initiated afterward. Docking steps are done. This file is carrying RMSD table, which is very important for us to obtain information which are quite important for the analysis of protein and ligand binding. So as we see in the first row, the ligand at the first row is the one with the best conformation possible and the best binding affinity. So we put the run number in the autodoc program, and we obtain further information, including all the necessary data and also the visualization of binding of the ligand with the protein itself. Of course, after we download the final result, after the analysis step, we can import the file into protein ligand interaction profiler website, where we can visualize it in more details, the analysis step. Now here we see that we have the result table. So the first ligand library is on the top, the second ligand library results are at the bottom, and the cancer drugs to be compared with them are basically on the right side. We need to make sure or know that always the inhibition constant needs to be as low as possible to have the highest inhibition ability, which is necessary here. So then after I put them all in one table and sorted them, I put them in a plot. So here we see at the bottom, the ones at the bottom are the ones with the highest inhibition ability and the lowest inhibition constant. And as we go up, we see that the inhibition constant is increasing and the inhibition ability is decreasing. So the ones with the best inhibition ability here are nilotinib and the ones with on the top of it, the other ligands, and then imatinib and so on, as we see in this plot. Also for DDR1, we did the same process. I only shared the results of DDR1 here. So this is the results for DDR1, the same steps, same everything. And of course, the results of DDR1 are also shown here. We see that some ligands were actually of best or better inhibition ability, more than the cancer drugs. So here we see two ligands at the bottom, other than cancer drugs actually. And here there's an important visualization, we can say, or an important comment. So we have the first ligand at the bottom with that's ending with 72, and the one after it, right above it, which is ending with 48. These ones are of the best inhibition ability based on DDR1, but also they are present in DDR2 plot. They are also of the best, most or top 6 ligands in the inhibition ability, so they can be considered as promising for the inhibition for both DDR1 and DDR2 as well. So in conclusion, pancreatic cancer is a deadly disease with a complex cellular pathways promoting the quality of TME, causing it to generate anti-tumor immune responses, which result in its escape from cancer treatments. The only method that can be promising in this situation is to target receptors in the extracellular matrix to disrupt the pathways leading to the aggressiveness of this type of cancer. It can be said that the results found are promising in the matter of high inhibition ability with a low inhibition constant, since the target sites were sites that are active in collagen interactions. Also, as we mentioned previously, it is a necessity to mention that both the ligands, the ones ending with 72 and ending with 48, are common promising inhibitory agents for both DDR1 and DDR2. Finally, I would like to convey my deepest appreciation and thanks to all my professors beside my advisors, Professor Tuğba Bal and Professor Zihni Onur Çalışkaner, who helped me a lot throughout my presentation of this, throughout my work, let's say. This project would not have been possible without their efforts, supervision, and guidance. My thanks are also directed to the conference committee and members for giving me this opportunity to present my work. I thank you all so much. If you have any comment or a suggestion, please let me know. Thank you.